So, good afternoon, students. And today, since your periodic test is very near, and we have already completed the chapter States of Matter and Cell. So, today we will do a revision of cell. Okay. So, we will revise with the chapter cell. And if you have any doubts, we can get it clarified also during this online interactive session. So we will revise with the chapter cell, which is there in your PT1, the syllabus of PT1. Okay. So student, before starting with the chapter, let me also have some briefing, or let me know that what you have learned till now from the chapter. Can anyone say me how we can define cell? How we can define cell? Anyone? How we can define cell? How we can define cell? Just raise your hand so that I can unmute you. Raise your hand so that I can unmute you. Just raise your hand so that I can unmute you. Okay, Nandini, you can unmute yourself.
Sir, cell is the structural and functional unit of life.
Life, very good. Cell is a structural and functional unit of life. Very good, Nandini. So the cell is the smallest functional unit of life, and it was discovered by Robert Hooke. And how Robert Hooke discovered the cell? What was the briefing regarding the discovery of the cell by Robert Hooke? Anyone? So, just a yes, Nandini. Yes, correct. You have written the answer in the chat box by bark of the tree. Okay. Now, cell is called the structure and function unit of life because it carried out the basic processes, the basic function of the life. Now, just a moment. Yes. Students, as per our syllabus is concerned, cell, basically there are two types of cells, prokaryotic cells and eukaryotic cells. So based on the type of the cells, there are two types of organisms. One is a prokaryote, another is a eukaryote. Now, anyone, can you tell me what do we mean by the prokaryotic cell and what do you mean by the eukaryotic cell? One by one, the differences, let us study, because from the diagram, you can see the diagram I have highlighted for the prokaryotic cell and a eukaryotic cell. It is there in the slide. It is there in the slide. Now you can see a prokaryotic cell. Just a moment. Yes. Prokaryotic, the word pro means primitive and karyon means nucleus. So those organisms with the primitive nucleus are called prokaryotes, and those organisms with the modern nucleus, eu means modern and karyon means nucleus. So those organisms with the modern nucleus is called the eukaryotes. Okay. So in prokaryotic cell, you will find the cell wall. This is a flagellum. And the most important thing is that the nucleus is not well defined. The unorganized nucleus is called, a special term is given to it. It is called the nucleoid. Okay. On the other hand, the important cell organelles are also absent in the prokaryotic cell, as we can see with the diagram, which is highlighted in the image. But these cell organelles are definitely present in the modern nucleus, that is a eukaryotic nucleus. So these cell organelles are present in the modern nucleus. So here you can see the cell organelles which are over here. Nucleus, a well-organized nucleus is there. Well-organized nucleus means the nucleus will remain surrounded by a nuclear membrane. Then the different cell organelle like mitochondria, chloroplast, Golgi body, ribosomes definitely present, and the endoplasmic reticulum. These all are present in the eukaryotic cell. So the unorganized nucleus is found in the prokaryotic cell, while in eukaryotic cell, the organized nucleus is present. Now, can anyone just can anyone differentiate between prokaryotic and eukaryotic cell? Can anyone differentiate between prokaryotic and eukaryotic cells? Any of the student? Can you differentiate between prokaryotic and eukaryotic cell? Anyone? Can anyone differentiate between Okay, Ritika, good. Unmute yourself, Ritika.
Yes, sir.
Yes, Ritika,
Sir,
answer.
prokaryotic cell have a well-defined nucleus and sorry, sir, not well-defined nucleus. And
Very good.
eukaryotic cell have well-defined nucleus.
Okay, good.
Second,
Okay, okay, okay.
prokaryotic cell.
Okay, Ritika, stop. Only one difference from your side. Next, I would like to ask, yes, Ayush. Ayush, unmute yourself, Ayush. Ayush,
Yes,
unmute. Ayush, apart from the
Yes, sir,
point which Ritika has said, any other point from your side?
Yes, sir. Cell organelles in prokaryotic are absent and cell organelles in eukaryotic are present.
Okay, okay. Very good, Ayush. Next, apart from this difference, let me ask some of the students. Okay, Shamli, unmute yourself, Shamli. I've given you the option, unmute yourself.
Yes, sir.
Shamli, from the two difference which Ritika and Ayush has shared, apart from these two difference, any other difference between prokaryotic and eukaryotic cell?
Sir, chromosomes in prokaryotics, chromosomes is single in number, whereas in eukaryotics there are more than single chromosomes.
Very good. Single chromosome in the prokaryotes and more than one in eukaryotes. Very good, Shamli. You can mute yourself. So, student, the three most important difference has just been shared by our students. That is, well said, that prokaryotes have they prokaryotes have unorganized nucleus. Eukaryotes have the organized nucleus. From the diagram also, which is there on the slide on your screen, you can see this. The labelings of the diagram you can get very well. We are not naming the nucleus of the prokaryote as nucleus. We are calling it as nucleoid. And true nucleus is present in the eukaryotes. On the other hand, cell organelles, important cell organelles, or we can say all the cell organelles are absent in prokaryotes, but they are definitely present in the eukaryotes. And chromosome single and eukaryote more than one. And regarding the size, prokaryotic cells are small and eukaryotic cells are definitely larger in size. Okay. Yes, correct. Okay, now we are going to the next portion. Let me allow some of the students, they have joined. Okay. Next, we are coming to the similarities and the differences between the plant and the animal cell. Okay, a very common similarity is that they both have the nucleus, cytoplasm, plasma membrane, that is a cell membrane, Golgi apparatus, mitochondria, endoplasmic. No problem. So students, regarding the similarities we have discussed, now can anyone tell me the differences between the plant and the animal cell? Just raise your hand so that I can unmute you. Differences between plant and the animal cells. Okay. Okay, Ayush, again, this time. Okay, this time Manish, unmute yourself, Manish. Manish, you can
Yes,
unmute
sir.
yourself. I have given you the option.
Yes, sir.
Unmute yourself, Manish. Tell me the answer.
Sir, cell wall is present in plant cell or cell wall is absent on animal cell.
Other difference?
Sir, ये plastid are absent in animal cell or plastid are present in plant cell.
Okay, good. Plastids are present in plant cell and absent in animal cell. Okay.
Cell.
Okay. Fine, Manish. Next, let me give other student also the chance. As student, it should be completely interactive because this I am revising because you have in your periodic test this chapter, so we are doing revision over here. Next, Abhinav. Abhinav, unmute yourself. I would like all the students to interact. Abhinav, unmute yourself.
Yes, sir.
Abhinav, continue with the differences.
Plant cell have very large vacuoles and animal cell generally not have vacuoles or present in very small size.
Very small. Okay, very good. Good. Okay, fine. Okay. Now, students, we are coming to our next portion regarding the cell organelles. Okay. Cell organelles. Now, the cell organelles, the first and the most important cell organelles we'll study is the cell membrane or the plasma membrane. As student, from the diagram which is depicted over here, structurally you can see in the diagram which is highlighted over here on the screen. So plasma membrane is actually the lipid bilayer. Okay, the lipid bilayer. So if you are talking about the structure of the plasma membrane, let me go with the text. It is lipid bilayered. Lipid bilayer. That means two layers of lipid is there. These are the lipid molecules. Just a moment. Yes. Just a moment. These are the lipid molecules which are, just a second. These are the lipid molecules, the two layers on the top. These are the lipid molecules. And the most important thing is that in between the lipid molecules, what is inserted in between the lipid molecules? In between the lipid molecules, the protein are inserted. Okay, the proteins are inserted in between the lipid molecules. So the plasma membrane is a lipid bilayered structure. This model is called the fluid mosaic model, and it was proposed by the scientist Singer and Nicholson. So the plasma membrane is an organelle which definitely separates the cell from its content. It acts as a boundary. It is composed of, as I already told you, lipid and the protein. And the most important thing is that plasma membrane is called now selectively permeable membrane. Student, previously what plasma membrane was called? As I told you during the classroom also, that plasma membrane initially the scientists called them by other term. And nowadays we don't use that term, we use the refined one. What is that called? Anyone? Previously, we were naming plasma membrane by anyone? Anyone? Previously? If you want, you can respond through the chat box also. Students, am I audible to you? Okay, yes. Previously, what the plasma membrane was called by? Anyone? No, Manish, it is his common name. I am telling you. Previously, plasma membrane was called by semi-permeable membrane. But nowadays, we are not using that term. We are saying it is selectively permeable as because it allows the flow of the limited material through it. Okay? Is it okay to all the students? Okay. Okay, so let me just recall once again. So can anyone tell me the structural component of the plasma membrane? Anyone? What is the structural component of plasma membrane? Raise your hand. Okay, let me ask. Okay, Ayush, unmute yourself, Ayush. Unmute yourself, Ayush.
Yes, sir.
Ayush, what are the two components of the plasma membrane? Plasma membrane is made up of? Yes,
Sir, lipid bilayer.
okay, lipid and? Yes, Shamli Ritika has given the answer through the chat box. You can also raise your hand so that I can unmute you. Yes, correct, Shamli Ritika. It is lipid bilayered and protein. Very good. Okay. Now, can anyone tell me the function of plasma membrane? Please raise your hand so that I can unmute you. It should be an interactive class. Nandini, very good, very quick. Nandini, please unmute yourself.
Yes, sir.
Tell Nandini.
Sir, it maintains the individuality of the cell.
Okay.
It protects the cell from injury, and plasma membrane is selectively permeable because it allows the entry and exit of selected material.
Very good. Okay. Correct. Next, Ritika. Ritika also raised her hand. Okay, Ritika, you want to answer? Answer it. Ritika, unmute yourself.
Yes, sir.
Tell. Ritika.
Sir, it controls the entry and exit of the cell.
Entry and exit of the cell or materials.
Yes, sir. Selected material.
Selective material into and out of the cell. Very good, Ritika. Okay. Let me ask randomly some of the students, those who have not answered. Yes. Shubhali, unmute yourself, Shubhali.
Yes, sir.
Shubhali, tell me the function of plasma membrane.
Sir, it maintains the individuality of the cell.
Okay.
It
Next.
protects the cell from injury.
Okay.
Plasma membrane is selectively permeable.
Yes, the
Membranes
most
allow the
important.
entry and exit of the cell membranes.
Okay, very good. Very good, Shubhali. Okay. Now, students, we are going to the next cell organelle. But before going to the next cell organelle, let me discuss some of the process, diffusion and osmosis. So diffusion definitely is applied for the gaseous medium, and it is a process by which the gases shift from the region of the or move from the region of the high concentration toward the region of low concentration. Osmosis is applied for liquid medium. Okay, definitely from the high concentration toward the low concentration. through a semi-permeable, or we can say selectively permeable membrane, but this is applied for only the liquid molecules. Now, students, we will come to, okay, but before coming to this topic, let me ask you that how much you can recall regarding the diffusion and osmosis. Can anyone tell me the difference between diffusion and osmosis? Just raise your hand. Okay, Ayush, very quickly. Okay, Shamli, Nandini, okay. Okay, Shamli, first I'm giving chance to you, Shamli. Unmute yourself, Shamli.
Yes, sir.
Speak.
सर osmosis और diffusion में ये अंतर होता है कि osmosis is the movement of water molecules from high concentration to low concentration, whereas diffusion is the movement of gases molecules from high concentration to low concentration.
Okay. Okay, fine. Very good, Shamli. Yes, you can mute yourself. Okay. Again, I think 1 minute. Just a moment. Okay, fine. Now, students, after this, we are coming to the very important part, that is the 3 type of solutions. Let me clear the drawing. The 3 type of solution: hypertonic, hypotonic, and isotonic. Now, see what are these? We were discussing about the different type of solution, the hypertonic, hypotonic, and the isotonic solution. Now, for this, let me use this image so that so if the amount of the water present in the medium surrounding the cell in which the cell is kept is same, then there is no net movement of the water molecule and the medium. We will be saying this to be the isotonic. Okay, when there is no net movement. But if there is the condition like, see, the most important thing, the medium surrounding the cell, if has the higher water if it has a higher water concentration than the cell, then the water will go inside the cell will swell up or inflate and eventually may burst also. So here the water is transported inside the cell, and the medium surrounding the cell has higher water concentration than the cell. This type of solution is called hypotonic. So can you tell me, student, that whether in this situation endo-osmosis is occurring or ex-osmosis is occurring? Anyone? In case of the hypotonic solution, what we are discussing. Okay, Nandini, very quickly. Very good. Yes, unmute yourself, Nandini.
For endo-osmosis.
Very good. It is endo-osmosis. I think Ayush also, you also have the same answer. You can unmute yourself, Ayush.
Yes, sir.
Okay, good. Fine. Correct.
Yes, sir, same answer.
Both of you are correct. And now, students, we will be going to the next type of solution, that is hypertonic. So in case of the hypertonic, it is opposite to that of the hypotonic. The medium surrounding the cell has lower water concentration than the cell, and as a result, the water from the cell will go into the medium surrounding the cell. So in this case, the cell will shrink. Okay, so this type of solution is called a hypertonic solution. And now, Abhinav, unmute yourself, Abhinav. Abhinav, unmute yourself.
Yes.
Abhinav, can you tell me
Yes, sir.
here endosmosis is occurring or exosmosis is occurring? Here in this solution,
Exosmosis.
very good. Here in this solution, exosmosis is occurring. Good. So at last, can anyone differentiate between the three type of solution? Please raise your hand. This I would like to ask all the students as because this is very important question for exam purpose also and for all. You can see because the three type of solutions are very significant for the plasma membrane during the study of the plasma membrane. Yes, one by one, this I will ask from all the students because this is very important part. First, okay, first Ayush raised his hand. Very good. Unmute yourself, Ayush.
सर हाइपोटोनिक में ये हाइपरटोनिक में सेल श्रिंक करेगा और आइसोटोनिक में नॉर्मल रहेगा और हाइपोटोनिक में सेल स्वेलअप हो जाएगा.
Okay, okay, correct. Ritika, unmute yourself. Ritika, unmute yourself. Yes, Ritika, you can unmute yourself. I have given you the option.
Yes, sir.
Differentiate between the three solutions.
Sir, in hypotonic solution, the concentration of water is more in medium than in cell, and
Okay.
in hypertonic solution, the concentration of water is more in cell than in medium. But in isotonic solution, the concentration of water is equal in cell and in medium.
Okay, fine. Very good. Next. Bharti. Bharti, unmute yourself. Unmute yourself, Bharti. Unmute yourself, Bharti. I think got disconnected. Okay, no problem. Shubhali,
Yes, sir.
Shubhali, differentiate between the three solutions.
Sir, in the hypertonic solutions, the water enter the in hypertonic solution, water is the lower concentration of water. Then the water enter in the cell and it swell up.
Okay.
It shrink. It and the hypotonic solutions, water enter the cells and it swell up. And the isotonic solution, cell is placed medium that has exactly the same water concentration as the cell. There will no movement of water across the cell membranes. Such solution is known as isotonic solution. This will stay the same size.
Very good. Very good. Well said. Well explained. Now, Nandini, unmute yourself, Nandini. Nandini, unmute yourself.
Yes, sir.
Yes,
Sir,
Nandini, differentiate.
Sir, in hypertonic solution, water moves out of the cell, and hypotonic solution, water moves in of the cell, and isotonic, there is no net movement. And hypertonic solution, the cell will shrink, and the hypotonic cell will swell up, and isotonic cell will normal.
Very good. Very good. Yes, correct. Nandini, very well said. Next, Manish. Unmute yourself, Manish. Manish, unmute yourself.
Yes, sir.
Manish, tell me the difference between hypotonic solutions.
Hypertonic, sir.
The three type of solutions, tell me the difference between the three type of solutions. Hyper, iso, and hypotonic.
In hypotonic, the medium surrounding the cell has higher water concentration, and then cell will gain water and it will swell up.
Okay.
In hypertonic solution, if the medium of surrounding the cell has lower concentration, then cell will lose water and it will shrink. Cell
Very
in
good.
isotonic, the medium surrounding the cell has equal concentration of water with respect to cell, then there is no movement of water molecule.
Okay, very good. Very good, Manish. Very good. Well said. Okay. Now, students, we are coming to one very important process, that is plasmolysis. You can see, student, in this image very well that plasmolysis, shrinking of the protoplast from the cell wall. Okay, because the cell wall being rigid, so the cell membrane will shrink. Okay, and as a result, there is a shrinkage. You can see in the diagram, this diagram clearly indicating this is a normal condition, deplasmolized cell, and here you can see it is receding out from it. The plasma membrane is shrinking, receding out of the plasma membrane. So this type of solution is called. Okay. Yes, Ayush, I have seen your query. You have asked to explain endocytosis also. Okay, Ayush, I'm coming to that point also. I'm coming. So what is plasmolysis? Ultimately, it is the process in which the plant cell loses water through osmosis, that means shrinkage. So when it is kept in a hypertonic solution. Okay. And now, after this, one more term which Ayush has just asked, though in the class also we have discussed, but here let me use the text to answer his question. Endocytosis. Okay, for this student, endocytosis is what actually, suppose this is the amoeba. Okay, suppose this is the amoeba. This is the nucleus of the amoeba. And if amoeba, this is a pseudopodia of amoeba. Okay, this is the pseudopodia. Now, if suppose amoeba get any, like, let me use with a different color. Suppose any food material amoeba is encountering, then the food material is engulfed. This blue color indicate the food material. So the amoeba takes in the food material through the plasma membrane by engulfing through the plasma membrane, and this process is called endocytosis. Okay, Ayush? Is it clear, Ayush? Okay. Okay. Yes, students. Okay, fine. Now, students, within few seconds, our class is going to end. So, student, today's assignment, definitely you will prepare the same chapter. And tomorrow, again, during the online class, I will continue with this chapter revision workout. Okay. So, thank you, students, for today's class.